Hello everyone, I am Ashik Mahmood. Thank you for joining with us to this ISCB-SC webinar. I would like to remind you, if you want to know about the upcoming webinars or how you can present your research and be part of this initiative, you can follow us on Twitter. Today we have an interesting talk by Mr. Ummesh Panwar, and he is a PhD student from Algapay University, India. If anyone have any questions, just give a comment in a comment box. Now please, Mr. Ummesh Panwar, start your thought.
Thank you, Mr. Ashik, for your nice introduction. Good evening, all. This is Umesh from Alapa University, Department of Bioinformatics. And I would like to thank ISCB webinar for giving me this opportunity representing my work. And today I'm going to present uh, about the virtual screening tool, identifying novel inhibitor for impeding the integrate LEDGF. So, before starting to the main concept, what exactly we have targeted over here, I just would like to tell you about that. Generally, the whole world is there are so many viruses throughout spread throughout the world. Specifically, you can see Ebola virus, diphtheria, MERS, SARS, HIV, cholera, plague, and chikungunya. Like that, so many viruses are over there. Among them, recently we all are uh, very much very much familiar with the SARS-CoV, uh, which has drastically taken the changes over the world. Uh, Among these, I just taken my work of my PhD work on HIV, so which is I'm going to talk about over here. Why I have chosen HIV because, as we all know, that 1981 the AIDS was first reported and HIV isolated in 1983. Uh, HIV infects generally the vital cells of humans, human systems like T cells, macrophages, and dendritic cells. And HIV is the member of genus lentivirus and part of the family of the retroviridae. This is the general structure of HIV. And if you see the current statistics of HIV in 2019, the 38 million people who are living with HIV in 2019, and almost 1.7 million people were newly infected. Among them, almost 69240 HIV-related deaths has been found in 2019. This was the statistics represented by WHO. And this is the general timeline of HIV from 1981 to till 2019. And if you can see how the HIV has started and what are the progress has been made throughout the last 20 to 30 years as of now. And there are many FDA approval medicines also came into the market, like from 1987 to Zidoviridine to till 2020, first remdesivir. These are specific FDA approved drugs are available in the market as of now. And why generally HIV is very important, and why I have targeted the specific HIV integrase here. This is the general life cycle of the HIV, where the life once the HIV enter into the human cells and inject their genomic material into the cytoplasm, and the further integration process will be done, and where RNA production and assembly process will make over the new budding and mature viruses which further infect the other cells. So if you can see the specific process in the nucleus integration, where the human DNA and viral DNA integrate to each other and produce for the new progeny. So what exactly we are going to have here? Is there any alternative option where we can control the viral infection? So what exactly I have done? I just tried to identify those specific cellular cofactors which are involving in the integration process, like human cellular cofactors, especially like LEDGF transprotein, SNU protein, MCM7. Like there are some other cellular cofactors are also involving. Among them, what we have identified through the literature, that the LEDGF p75, which is directly involved in the integration process and helping into the replication. So how this LEDGF binds to the integrase during the integration process. We can see over here. This is the general process of targeting the LEDGF, targeting the integrase by LEDGF. So where the LEDGF is having three different kind of domain. One is PWWP, HE hook, and IBT. And where the IBT domain, integrase binding domain, binds to the integrase dimer interface, and further start the application process. So how this interaction looks like exactly, where these are the key interacting residues where the integrase CCD catalytic core domain binds with the IBD of LEDGF. The specific residues from the CCD was identified with, through the experimental studies was the tryptophan 125 and threonine 125, tryptophan 131, and glutamine 170, histidine 171, and threonine 174. These were the specific five residues has been identified to have the interaction with the IBD. So based on these interaction, what exactly we have, we are trying to find out the new molecule which can block their interaction. So this is our aim to identify those specific new molecules from the chemical library which are already existing, and we are going to block their interaction. How those new molecule can mimic exactly like LEDGF to bind with integrase to their specific residues where LEDGF used to bind. So what we have done, we have through one nature chemical biology one literature, we have taken the already known crystallized compound, and we have read about the information of their interactions and all. What exactly Christ has identified over here, they have identified a specific molecule which binds to the these glutamine 170, histidine 171, and threonine 174 of the integrase, and exactly replace the LEDGF for blocking their interaction with the integrase. So based on this information, what exactly idea we had over here, we just tried to find out those specific molecules from the specific libraries like specs and amine and uh, zinc libraries and other libraries also we have targeted. And further we went for the molecular docking and their evaluation through dynamics. So based on that, what exactly we find, we found the specific compounds, and we have compared our results with the known already known co-crystallized compound, where we found the enamine compound, maypris compound, and spec compound, three specific compounds through IFG, and where the docking score of the our results were having high with exactly similar interaction where the known compound was also binding into the integrase binding pocket of LEDGF. So based on this, how exactly we saw like okay, the compound is exactly binding inside the surface of the LEDGF binding site, is exactly fitting into the pose. So based on this strategy, what further we have gone for the enrichment calculation analysis and admit analysis. In the enrichment calculation analysis, what are the compounds we have rectified, we have searched, we have gone for the ROC curve generation, and where we understand that how the ROC score is giving a better model as per the already those decoy sets. Comparison to decoy sets, our compounds were coming on the top and giving a best result. So, and also we also identify their ADME properties and pain prediction, which represents the molecules with acceptable ranges, which has been already notified for the human ranges. And then further we've gone for the dynamic studies of those specific compound along with the known compound, where we identify the known compound, the coxless compound has lost its RMSD, and you can see how the deviations was identified after the 15th nanosecond. It has highest deviation, but as compared to our known new compound which we identify, almost the all three compound, especially the anamine and specs compound was most uh, stable throughout the simulation trajectory analysis, and we also identify how the compactness of this compound has been affected their gyration. So based on this study, what exactly we've identified that these three compounds from anamine and specs and Maybridge can be select for the experimental study, and we can go for the checking out their inhibitory activity via in vitro studies, where the experimental studies is in process as of now. So once the experimental data will be there, we are going for the comparison of the already known compounds, and we will further go for the what are the possibilities are there. So this is the general idea which we had over here. This is the very simple concept, and we have already the in silico already have published in the JBSD paper, and experimental work is in process. Once the experimental result will come, we will try to communicate that one also. So the general briefing was the identification of the new compound which can block the integrase and LEDGF binding, which will be helpful for the avoiding the replication process and integration process. So this is our general idea over here. And I just would like to acknowledge my supervisor, Professor Sanjeev Kumar Singh, other my lab colleagues, as well as Aruf, ICMR, and Rusa and GBT for giving me funding opportunity to work in the research laboratory. And thank you for everyone all. Thank you very much.
Thank you, Mr. Umesh Tanor, for your amazing
Thank,
talk.
thank you, sir.
I've seen that you have used molecular docking to block integration. Has there any
Yes, sir.
limitation of your study?
Yeah, we had sir limitations of the studies where we what we exactly we found out that the one specific residues which were involving in the binding site of integrase and LEDGF, that was uh, glutamate 168, which has not involved in the our study as well as the whatever the compound which we have taken. So in the next while targeting the next work, we have tried to identify how these specific residues are involving in this particular portion, and whether it's important or not. Based on that, we gone for the dynamics study specifically for those specific residue, and that work is still in communication.
Okay, okay. So any questions from audience? Please write in comment box. Okay, I think there is no question.
Thank you, sir.
Uh, it was a really amazing talk. And thank
Yeah,
you, Mr. Umesh Tanor, for presenting the webinar.
yeah, thank you. But sorry for that. I was not knowing about that timing. I was very short presentation
No,
only
no,
I have prepared.
it was a really wonderful presentation and really easier to understand. I think audience will really understand your presentation very easily. And for
Okay.
a small reminder to all, we arrange two webinars per week, so stay connected with us. Good day and stay safe.
Sure, sure, sure. Thank you very much, Ashik sir.
Okay, Mr. Umesh Manoj, thank you.
Thank you. Take care of you.